Our next speaker is Pravin Dugal, and the topic is subretinal human retinal progenitor cells in retinitis pigmentosa, a phase 1/2A study. Please.
Thank you, and good morning to everybody, and thank you to the organizing committee. Thank you, Suzanne and Vas, for allowing me this opportunity. My financial disclosure of note is that I'm a consultant to Renuron. Renuron's retinal progenitor cell, or HRPC therapy, consists of cells that are isolated from the fetal retina, can differentiate into retinal cells, and can be cryopreserved with a 9-month shelf life, and no immunosuppression is required. The mechanism of action is both integration and paracrine effects that may contribute to its efficacy. The potential benefits of subretinal HRPC include direct delivery into the subretinal space, on-demand shipment to site of care, and perhaps most importantly, treatment that is agnostic to genetic subtype of disease. The first disease that was targeted was retinitis pigmentosa. A phase 1 safety study has been done, and this consisted of recruiting 12 patients with severe disease who were given 3 different doses of the cells. This data was presented by Dr. Jason Commander in ARVO. It showed a good safety profile and allowed for progression into a phase 2A study. And today, I'll be showing you for the first time the interim results of the phase 2A study. Here we recruited 10 patients with a baseline visual acuity between 20/640 and 20/80. There was a single dose, a 1 million cell dose in a cryopreserved formulation, and the primary endpoint was a change in ETDRS letters read from baseline to 24 months post-treatment, and 2 sites were involved. The procedure itself consisted of a core vitrectomy followed by creation of a subretinal bleb and then injection of the cryopreserved cells through a small gauge needle, as you can see in this video. I'll first show you the safety results of all patients. In both studies, a total of 22 patients were treated. The dose escalation was generally well tolerated. There was no evidence of inflammation or proliferative vitreoretinopathy. There were 2 ocular SAEs that were reported that were not felt to be related to the drug product. That included a progression of a pre-retinal epiretinal membrane requiring additional surgery, as well as persistent subretinal fluid with a patent retinotomy. There were 2 events leading to loss of vision related to either and or the surgical procedure or the patient's selection. One was an RPE tear and the other was persistent subretinal fluid with a patent retinotomy. And now I'll show you the visual acuity results. And you can see that in some patients there was an immediate and dramatic improvement of vision, and in others there was a slower improvement. However, as you can see as circled in red, there were 2 patients who lost vision. At the 90-day mark cumulatively, there was an improvement of 6.1 letters of vision in 8 patients. At 180 days, there was an improvement of 18.5 letters of vision in 4 patients. Now, it's important to state that because this was an open label study, the fellow eye that was not treated always has to be considered. And in the same time points at day 90, there was an improvement of 6.8 letters of vision in 8 patients, and at day 180, there was an improvement of 7.8 letters of vision in 4 patients. If we graph the treated and untreated eye, you can see that the largest separation or delta occurs at day 180. If we exclude the 2 patients who lost vision due to the surgical procedure and or patient selection, you can see that the delta exists throughout the course of the study. So in summary then, there appeared to be an acceptable safety profile. There were biological efficacy signals that were very rapid and profound in some patients and slower in others. These study results will provide a better understanding of the optimal patient selection as well as surgical procedure standardization for future study design. And finally, this is potentially a promising new therapy for patients with retinitis pigmentosa for whom a few therapeutic options currently exist. Thank you for your attention.